심포지엄 이제 곧 시작할 텐데 제가 하고 있는 연구가 기후변화 대응 가축유전자 연구라는 한국연구재단에서 지원하는 기초연구실 사업을 수행하고 있어요. 연습이었어요. It was a practice that what I'm going to speak. 두시되면 뒤에서 알려주실 건가요? 거기서 시작 신호를 주시면 그때부터 시작을 할게요. 네, 그럼 지금으로부터 가축유전자 대응 기후변화 대응 가축유전자 연구 심포지엄을 개최하도록 하겠습니다. 제가 지난 3년 동안 한국연구재단 기초연구실 사업을 통해서 가축유전자 연구를 해왔는데요. 그 연구에 도움이 될 수 있는 분들을 모시고 오늘 심포지엄을 개최하게 되었습니다. 오늘 심포지엄은 두 개의 세션으로 나눠져 있는데요. 첫 번째 세션은 개발도상국 축산업을 위한 협력이라는 제목으로 아이와 주립대 로스차일드 교수님과 코쿤이라는 업체의 양희재 본부장님이 발표를 해주실 것이고요. 두 번째 세션에서는 생명공학 기술의 활용이라는 주제로 캘리포니아 대학 축산학과 알리슨 반 에넨남 교수님의 발표와 그리고 서울대학교 수의과대학 장구 교수님의 발표가 있을 예정입니다. 그럼 오늘 첫 번째 발표를 해주실 아이오와 주립대 맥스 로스차일드 교수님을 소개하고자 합니다. 제가 1997년부터 2003년까지 아이오와 주립대학교에서 석사와 박사 과정을 공부했었는데 그때 저를 가르쳐 주신 지도 교수님이시기도 하고 졸업한 후에는 또 제가 몸담은 가축 유전 육종 분야에서 제가 성장할 수 있도록 보살펴 주셔서 지금의 저를 만들어 주신恩인이십니다. 아이오와 주립대 맥스 로스차일드 교수님의 동물 유전학 분야에서 많은 중요한 공헌들이 있었는데요. 그 일부를 소개해 드리면 아이오와 주립대학교에서 1997년부터 2001년까지 동물 유전 육종 분야 책임자 있었고 2002년부터 2012년까지 동물 유전체 센터 소장을 하셨고요. 2012년부터는 글로벌 푸드 시큐리티 컨소시엄과 센터 포 서스테이너블 루럴 라이브리후드라는 프로그램의 책임자로 활동하고 있습니다. 특히 1993년부터 2013년까지 20여 년 동안 미국 대지 유전체 연구 프로그램의 리더로 활동하시면서 미국 내 가축 유전체 연구의 활성화와 성과를 공유할 수 있는 엔젠맵이라는 플랫폼을 만들었는데요. 이 플랫폼은 오늘날에도 전 세계 동물 유전체 연구자들에게 가장 활발한 토론과 소통의 장으로 이용되고 있습니다. 로스차일드 교수님은 매년 우간다와 탄자니아와 같은 아프리카 국가의 농촌 지역을 방문하시면서 동물 유전, 가축 생산 및 국제 농업에 대한 전문 지식을 공유하고 지역 주민들이 자연스럽고 풍부한 동물 유전자 자원을 선택하여 사용할 수 있도록 돕고 계신데요. 오늘 그 내용을 들어보고자 합니다. 자, 맥스 로스차일드 교수님을 소개해 드리겠습니다. 박수로 예.
안녕하세요. It's a great pleasure to be here today. I hope you all can understand my English, so I'll speak slowly. But it's a great pleasure to be in Korea again. I have visited your country many times, nearly 15 or 20 times, and it's always a pleasure to meet and visit with you about the things that we have in common and that we work on together. So today I want to talk about livestock genetics and genomics and how to improve food security in the developing world. And so what I wish to do to start is to remind you of the problem. In Korea you have lots of food, and it's very good food, But in parts of the world, that's not true. Perhaps 825 million, or about 10% of the world's population, suffers from hunger on a chronic basis. And more than 3.5 million children per year die from hunger. And this problem is made worse, much worse, by the war in the Ukraine right now, because that controls some of the food that's shared around the world. We don't depend on food only in our own countries, but we depend on food that's produced around the world. So what are the impacts of malnutrition? Well, reduced immune function. Children, their cognitive and physical development is impaired. They're stunted. Work productivity, lifespan, quality of life is poor. And what's even harder to see is that when we have hunger, when people go hungry in many countries around the world, then all the disease issues are worse. HIV, malaria, and it robs people around the world of human potential. We have to be citizens of the world. We have to help. And so one of the things we need to think about is how to reduce hunger. And one of the things one sees in developing countries is that when people are hungry, they tend to use land that shouldn't be farmed, shouldn't be torn down and plowed. And hunger also fuels social conflict and war. So what's our challenge? Well, by the year 2050, when all of you will be alive, old guys like me, we won't be around, but by the 2050, there'll be nearly 10 billion people in the world. So we somehow have to produce more food. And most of that increase in people will be in the developing world, not in places like the US or in Korea. But we need to really produce perhaps somewhere between 70 and 100% more food. That's the estimates, if we're going to produce a world in which children aren't hungry. We also have to do this with sustainability. And we all know about climate change. We have more drought, we have more higher temperatures, lower temperatures, unpredictable climates. This is particularly true in Iowa, where I live, where half the state now is having drought. Or in California, where I used to live, where the state is undergoing, I think, a 20 or 30-year record drought. So we have limits to water and energy and labor, and they're all required to produce more food, and we have to diversify our systems to make this work. So now I want to come to what role we can play, all of us, because you're animal science students, so we should be thinking about how we can play a role. And in many parts of the world, animals are crucial to economic survival, especially in developing countries. Animal source foods provide critically needed micronutrients and proteins. They serve as an economic bank. People hold on to animals because they have value, and they can sell them when they need money. In many countries, especially in the developing world, animals produce about 50% of the agricultural gross domestic product. And, which we'll talk about later, locally adapted animals are good for disease resistance, but they're not very productive. So genetics and genomics can be used to improve them. So this slide is a bit old, and I'll update it in a moment. But as you can see, the demand for livestock products, meat, milk, eggs, has been increasing ever since it's been increasing for the last almost 30 years. But more importantly, that increase is mostly in the developing world. Countries of Africa, Latin America, parts of Asia that are poorer than Korea, for instance. And if we look more recently, in terms of the growth of meat production by region and meat type, the dark blue, which, see the pointer, you got it. Well, as you can see here, the dark blue spots, these relate to the increases in production, that's the demand in the developing world. So both for beef, pork, and poultry, less so for sheep meat. Most of this increase that's expected between 2017 and 2029, really over the last, the previous few years and the next 7 years, this demand is mostly in the developing world. So almost all the demand that's coming forward in the future is in the developing world. So we need to produce more animal products, and genetics and genomics are key to that production. So let me just give you a little bit of history for just a moment, if you'll allow me. The first improvement to livestock began with domestication, which occurred, depending on what history and which species we work with, somewhere between 9,000 and 12,000 years ago. And domestication allowed for a more stable animal food source. You didn't have to go out and shoot that mastodon. You could raise your own cattle, you could raise sheep and pigs and chickens, and that would produce more food. And then about 350 to 400 years ago, breed development happened. So we started to, within a species, develop specific breeds of animals that fit certain needs. And more recently, we call it what happened in modern animal breeding and genetic improvement, perhaps happened over the last 75 to 100 years. And this included first selection based on the animal's appearance, phenotypic selection, and then later we developed methods to genetically evaluate animals based on performance of their own records and their records of their offspring and their relatives. And finally, we helped to develop delivery systems, artificial insemination, embryo transfer, and now more recently, the use of sex semen. Based on my age, I came in here somewhere at the start of the genetic evaluation system, or the improvement of the genetic evaluation system. More recently, we have what's called the genomic revolution, which occurred 25 to 30 years ago, where we look at sequencing and gene identification itself. All of these are ways to improve livestock. And if you look at the success we've had, and these are trend lines for dairy cattle milk productivity from Europe, New Zealand is the bottom line, Canadians and the US, you can see that there have been enormous improvements. Now, you might ask yourself why the bottom line isn't increasing as much. Almost all of that is grass-fed. But even so, that's an improvement of over almost 40% in terms of milk production over the years. So we've made enormous improvements, and we can show the same in pigs, in chickens, and other species. Why the success? I always ask students this. Why do you think we've been so successful in animal production? And most students tell me, well, it's genetics, or it's artificial insemination. And in fact, the real reason, the first reason that we should consider is we finally provided animals with unlimited feed and water. All the other things, improved health, improved management, sophisticated animal genetics and genomics like you've learned in your classes, they allow the genetic potential to be realized. But it starts with more feed and more water and more care for the animals themselves. And if we look in the developed world, and Korea is the developed world, we don't yet have barcodes on our animals, genetic barcodes, but we're getting very close. We can almost take a sample of DNA and tell you exactly what kind of performance we expect that animal to have. But if we consider that, that's really just for the developed world. And it's for large animal production systems, much like you have here in Korea. I've been in the country now a few days, and we've seen some cattle operations. You don't have cattle grazing. You have cattle in buildings where they're under tight control. They're being fed. You know their genetic potential. But that does not happen in the developing world. In many places, for instance, in Africa, where I've worked a lot, they don't have enough feed or water. They don't have any way to cool the animals. They have very few facilities. They have no fans. They have no drip cooling. Some of them have shelters to provide shade, but not all. And herd size is so small, it makes traditional genetic improvement very, very limited. So what role do genetics and genomics play in developing countries? You're taking some genetics classes, maybe a genomics class. How are they going to play a role in improving livestock in the developing world? And the answer to that is we can use genomics to find the genes that are associated first with diversity of phenotypes. Second, to find genes that are associated with the animal's ability to adapt and be sustainable. And finally, we can find genes to improve performance and health. And what I'm going to do is show you a little of these examples today. I'm going to talk about the use of genomic selection. One of the speakers will speak on gene modification later today, so I won't really say much about that. But once we find those genes, we can use either genomic selection or gene modification. So I want to cover some of these topics for you today. And the first I want to start with is genomic diversity of livestock. So what does that mean? Well, it means that we screen animals to see how genetically diverse they are. And that's a key then, that when we go to select the best animals, there's genetic variability to use to select upon. This was originally done using microsatellites. The Food and Agricultural Organization recommended that. But in general, most of those studies were flawed. They used too few microsatellites, so we didn't get a very good picture of the genome. Now we suggest using single nucleotide polymorphisms, or SNPs. It would be nice if there were low-density chips because they would be cheaper to use. But in fact, many people in developing countries, scientists from there, they just don't have the availability of these types of chips, and it's too expensive. And genomic sequencing, which is the gold standard now, that's too expensive as well. One of my postdocs decided, well, at the time, there were no SNP chips to look at camels or New World camels like the dromedary, or Old World camels, or New World like guanaco or llamas. And so we decided to look at these animals using cattle chips, goat chips, and sheep chips, because they're all ruminants. So we hypothesized that these could be used to study the different camel species. And that's true, they can be. I picked just two articles to mention here. So as I said, we can do some sequencing, and the paper on the top, refining the camel myostatin gene polymorphism. This is a paper that your professor, Dr. Kwan-Suk Kim, did work on. This is by sequencing. Or you can use SNP chips. The lower paper is an example of looking at divergent selection in cattle breeds. Again, this is work that Dr. Kim and his colleague Hee-Jae Kim, who was a postdoc in my lab, worked on. So these are ways these are examples of how scientists, primarily from the developed world, are looking at genetic diversity in animals in the developing world. One of the things that we really need, one of the tools that we really need, is one chip that would have maybe 10 or more species and have SNPs from all these. And then that chip could be used very cheaply by people in the developing world and people in the developed world to screen for genetic diversity. So they might have 5,000 SNPs across 10 or more species, and they could be used rather routinely. Well, what about the genomics of adaptation? Can we identify genes or gene families that allow animals to live and prosper in very diverse climates? And here's a picture of water buffalo and cattle on the left from Egypt, and some fat-tailed sheep actually from Egypt. One of the things most interesting to scientists and others is what climate and change and heat stress does to livestock production. If we look at livestock production, heat stress really is important in terms of cost. It reduces productivity, it increases death losses, it ruins product quality in many cases, and it makes it difficult to keep animals healthy. In the U.S., the estimate is over $4 billion per year. In Iowa, where I live, in central part, it gets extremely hot. We have losses of chickens, cattle, pigs, very expensive. In global agriculture, heat stress probably causes over $100 billion US a year. Now, you might say that it should be easy to reduce the problems due to heat. We should be able to mitigate that. But in developing countries, the small stakeholders lack any of the resources to provide cooling technologies. They might provide shade, but no fans, no drip cooling systems. So very difficult in developing countries, quite different from what we see in the developed world. So heat stress affects global food security. And one of the things that geneticists like myself and Professor Kim have been working on are, can we identify traits and the genes that underlie those that affect heat stress or drought tolerance or disease resistance? So if we could identify those genes and mutations, then we could breed resilient livestock and produce animals that are more productive under these conditions. At Iowa State, we're working with a number of different groups to look at cattle, sheep and goats, poultry, and pigs. And we've actually done work here with pigs in Korea looking at heat stress. And these are some of the projects that I'm going to talk briefly about a few of them. One of the projects that I've been interested most in are sheep and goats, which are extremely important in the developing world, and understanding why some of these breeds do well in very hot and difficult climates. So these are pictures from Egypt, where it's extremely hot. There's virtually no rainfall, and lots of solar radiation. Grazing is on very poor pasture. And the question might be, how do these animals survive? And more importantly, if we find the forms of genes that make them survive, do those genes and the beneficial alleles, the beneficial form of those genes, do they exist in the developed world in some of the breeds that we have? So my research group has looked at a number of different goats and sheep in Egypt. These are some pictures of them, very different phenotypes. You might ask yourself, how do these animals survive in such hot and stressful environments? And we've been lucky enough to receive funding from the Illumina Greater Good Grant to look at the genomics of these animals. This is just one example of a paper that was authored by Isu Kim, who was a postdoc of mine, to look at signatures of selection in goats and sheep from a variety of hot and arid climates. Now, I don't expect you to completely understand your undergraduates. I don't expect you to understand what some of these graphs are, but what I want to show you today is that two things. The first is that there's a variety of ways to study and look for genomic regions, parts of the genome, that are associated with the traits of interest. And these are three approaches, FST, RSB, and IHS approaches. They all come up with slightly different answers. But here's just to show you a summary so you get a feel for this. In sheep, they suggest that there were somewhere between 7 and 13 regions that are associated with a tolerance to heat and these difficult environments. And in most cases, not all cases, they find the same regions. So we use different methods to find regions of interest. What's particularly interesting in this study was a comparison of what we found with goats and sheep. So I remind you, goats and sheep evolved from the same general species millions of years ago, and their chromosomal regions are very similar. Goat chromosome 12 is almost exactly the same as sheep chromosome 10. And what we found is that both in sheep, in this region, and in goats, there's a region on goat 12 and sheep 10 that are associated with resilience and to heat stress. So different species, same genes, but they're both associated with the ability to withstand stress. And we're in the process of trying to understand that region more thoroughly. You can also look at climates across the world, and they use something what's called a Cocoon climate classification that rates areas to be, depending on the type of climate that exists, they rate them into 4 groups. Tropical is the A group, dry and arid. The temperate, which would be most of the U.S., and the continental areas, where there's microthermal climates. If we compare livestock production across all these countries here on the left, more than 20 countries, we can look at sheep and goats from these regions. In this case, it was goats, and we can compare them. And when we do that, what we see is that there are regions associated with environmental adaptation in the goat genome. So there's areas of the genome that are fixed, years and years of selection, natural selection, and artificial selection by local producers to produce animals that were more stable. Then in those regions, we have genes that have been fixed in the populations to allow these animals to be more productive. And this is a paper, again, I chose one from Professor Kim that looks at diversity in Ethiopian sheep and looks at the different populations to find similar regions. One of the interesting projects at Iowa State, where I'm a faculty member, looked at crossing chickens from Egypt, the Fayumi, very small animal, with broilers, and then continuing to cross this year after year, and then to look at these genomically for regions associated with heat stress. And this is what's called a Manhattan plot. What we're looking for is regions that, again, are associated with, in this case, animals that are able to withstand heat. We did a number of testing of these animals, excuse me, for heat stress. And what we found were these regions that were associated with animals that could withstand heat and climate differences. And in fact, one of the interesting things in chicken, when we compare chickens across the world, chromosome 27 is kind of the African chromosome. It has a number of genes associated with the ability to withstand heat stress. This is work done by my colleague and her students, Sue Lamont and her students, in a project that I'm also associated with. There clearly has been selection for genes involved with feather development and heat shock. By contrast, in the northern climates, in northern Europe, chickens have been selected that have fixed genes on chromosome 2, that these genes are associated with the ability to withstand cold stress, the ability to be more sustainable in these types of environments. So natural selection and breeder selection has helped to fix certain genes in the population that are associated with productivity and, more importantly, adaptability. Probably the best known gene associated with heat stress in cattle is the SLIC gene. It's actually a mutation in the prolactin receptor. In the prolactin gene. And this gene can be found, it is now known in cattle. And it produces it produces animals, and here's an example, one with the mutation and one without. This allows these animals to be selected to withstand and be more productive in very hot climates. What's important about this, and I want to make a point about this, is if we're going to do genetic modification, we need to identify first genes or regions that are associated with the traits we're interested in. So somebody has to identify those first, and that's the role that genomics can play. What about genes affecting performance traits? One of the best known ones is associated with the Meishan pig. The Meishan pig is from China. It's known to produce as many as 40 piglets, but on average they produce 4 to 6 more piglets than most European breeds. Work done in my lab showed that the estrogen receptor gene is responsible for more than 2.3 pigs per litter in this breed. And we asked the simple question, does the beneficial allele in this gene, exist in other breeds? And in fact, it does. It exists in large white pigs, and it's also beneficial. Not 2.3 pigs, but about 0.8 of a pig per litter. So that's a large effect. That's about 30 years of breeding to produce. One gene can produce that much. So there are genes out there that not only are associated with adaptability and heat stress and sustainability, but also single genes associated with production traits. And we can find these looking at exotic breeds from different countries in the developing world. Well, if we can't find the individual genes, we can still use something called genomic selection. This is where we phenotype all the animals. So we phenotype all the animals, then we genotype them for at least 50,000 SNPs. Nowadays you can do 1 million or so. And then we can predict the phenotype based on those SNP genotypes. And we could do this repeatedly, and eventually, if we do it well enough, we can eventually skip doing some of the phenotyping, just use genomics. And this is particularly important in the developing world, because in the developing world, we don't often have the ability to phenotype animals easily. Well, where are we headed? We're headed to the situation in the world of a data revolution, where we can use sensors and cameras. Many farms are starting to use these. They could use sensors and cameras and smart necklaces, pedometers to see how far animals walk, and use all this to collect more information, both on sustainability and on ability to produce. And if we use that information, then we're going to have to use something called machine learning or artificial intelligence systems to harness all this genomic data and all the trait information, and to integrate it with field data and market information. So this is an enormous challenge. So for those of you that are interested in bioinformatics, you're interested in the ability to collect data using sensors or other cameras, this a whole new area for animal genomics people. Well, I want to finish now by at least getting you to consider, I've talked about the positive things. Now I want to consider some of the roadblocks, some of the difficulties that exist, because this is what requires bright young minds to help solve these problems for the future. So genomic selection really has great potential, because you don't need pedigrees aren't required to estimate breeding values with useful accuracy. This works best using data from the country you're working in. But there's lots of challenges. Initially, there's not an effective phenotypic recording system. And it requires a lot of funding to genotype large reference populations. And then finally, once I find that perfect cow, how am I going to deliver, or that perfect bull, how am I going to deliver that genetics and that genomics in developing countries that may not have good road systems, may not have people trained to use artificial insemination. So there's lots of issues around it. Lots of promise, but lots of concern. And there's other considerations, and this is where training young people like yourself is so important. We have institutional and human capacity building that's required. We have to consider gender. And we also should talk about policies that support livestock and related issues. So when you think about building up human capacity, if you're thinking, about raising a 1 year ahead, we say, well, let's just raise livestock. If we're thinking of 10 years ahead, we talk about developing breeding programs. And finally, if we're thinking about 100 years ahead, we should think about educating people. And so the role that you'll play in the future as educated animal scientists is to help train others around the world. So we need to increase the demand on science in livestock production. The days of just raising livestock are over. We need to integrate all forms of science into the process. We need more advanced training of young scientists from developing countries, and we have to encourage them to return home. Dr. Kim had a very bright young fellow from Ethiopia who worked here, was trained here, and then went back. We need to do more of that. And in my lab, we've been trying to do that. And one of the things is we need to encourage people from around the world to do sabbaticals and to help establish researchers to help in developing countries. We need to consider technology and gender as important. In many countries in the world, culture prohibits women from participating to the fullest in agriculture. I can't speak for Korea except to say that when I first started coming here, I wouldn't see very many female students. But now, thankfully, women are more integrated into animal production and into agricultural production. But we can't let culture and tradition hinder technology transfer. It's been estimated that if women had access to all the inputs in developing countries, that would increase household yields by over 30%. It's very important. And this could reduce the hungry people by over 10%. We need to talk about improving institutional capacity. So institutions like your university here can help in that regard. We need to have better equity between plant and animal science. In the US, I've been complaining about this for years, but the government, I don't know about here in Korea, but the government in the US and in groups like the Gates Foundation, they only invest about 20% of their money into livestock and primarily into livestock health issues. We need a more equitable solution relative to livestock and plants so that animals can play a bigger role in developing countries. And we need to get governments to provide more support for animal genetics research in Korea, in the US, and in other countries around the world. So investment in facilities and research support and training. Finally, and I won't give this issue enough time, but finally, we need to talk about who decides these policy issues. There are no silver bullets. We talk about silver bullets as ways to solve things. There's no silver bullets for developing countries. Sadly, many people in the developed world are telling the developing world how to operate. We need to have participation from all the parties. People in the developing world need to be given the chance to think about the benefits in their own countries. They need to have the right information to do so. And finally, all of us need to make a better case for biotechnology. Biotechnology is crucial to improving. Remember, I said we need to increase food production by 70 to 100%. Biotechnology is going to play a big role in that. So what have we learned in conclusion? Animal genomics can contribute to resilience and sustainability, but education and investment in capacity building are critical. Discovery work, application, policy, all this has to work hand in hand. And we need to have smallholders, the people that produce the livestock in the developing world, play a role. We have to employ the right tools depending on species, and we need to get the public and private sectors to work together. So, my future projections, eradication of food insecurity is going to require big innovations in animal agriculture. Genetic inputs can be optimized for local conditions and needs, so we need to work in developing countries. We can't force things that work in the developed world onto developing countries. That just won't be useful. And finally, we need to have new breeding technologies and digital tools that will allow tailor-made genetic development and dissemination of information. So our challenge is to feed a hungry world. And what I ask of each of you is to think about where you'll contribute. This is an enormous global challenge, and it really is something that we should all grasp. We can all participate in animal science, in animal production, in some of the outlying fields, veterinary medicine, biostatistics, to help produce more food in a hungry world. And I think this is a challenge that all of us, I think, will make us good citizens of the world. So I want to thank you, 감사합니다, and I'd be happy to answer questions if there's time. No? Okay. Thank you.
Q&A session will be available after the old session. We'll have a discussion time. 학생 여러분들께 좀 듣기에 이해가 안 되는 부분들이 많았을 건데 앞으로 그런 부분들 여러분들하고 또 공부할 수 있는 기회 가지고 싶어서 이런 자리 마련했고요. We'll have a second presentation from Hee-Jae, Mr. Hee-Jae Yang. Hee-Jae Yang graduate from Yonsei University, studied statistics. I had known him for 4 or 5 years since you work for the Cocoon company. Hee-Jae Yang will present his current project, Parasite research biobanking and cattle development in Tanzania. 잠깐만. 양희재 님은 그 코쿤이라는 충북에 있는 바이오 업체입니다. 거기에서 탄자니아의 기생충 은행과 그다음에 유구 개발 사업을 하고 있거든요. 백그라운드가 통계학이에요. 그리고 앞으로 그런 일들이 더 많아질 거니까 여러분들 좀 관심 있게 들어줬으면 좋겠습니다. 네, 감사합니다.
네, 소개 감사합니다, 교수님. 제가 같이 참석해 주신 패널 분들도 계시니까 발표 진행은 영어로 좀 진행하겠습니다. So before we move on, I would like to present a little bit about what Cocoon is doing, and later on I will introduce our two projects Professor Kim just mentioned. One thing is about the Parasite resource biobanking project which we are holding on in Tanzania, and another project we are preparing in Tanzania is cattle development project. So before we move on, I would like to remind you what is biobanking, because biobanking is what Cocoon is aimed to do. But by the definition, biobanking is a process of acquisitioning and storing activities related to collecting, preparation and preservation that I want to call manage, and analyzing and distributing biological samples as well as their related associated data. In the process of biobanking, I mean in the process of collecting, managing, analyzing and distributing samples and data, what Cocoon is doing is that we support research institutions, companies that you can use the biological materials as for their researches and R&D productions and manufacturing one. So here I will introduce you by one of our main software solutions. The first thing we stepped into the biobanking industry is with the IT solution. We called it Eenennaam. It's an integrated cloud-based biobanking information management solution that allows you to record any information related to your samples. So when you're in the lab working with the genetic analysis or working with the specimens that you want to add some research on, we now allows you to have a sample barcoding and monitor your repository yourself, and you can track easily the where is your samples at in our biobanking system. Long story short, we are providing our now systems to microbiology and cell line productions and of course parasitology. And we have another solutions that powered by artificial intelligence, so which we help researchers to identify the parasites in the samples itself. And I will gonna skip the details because later on I want to more focus on what we are doing in Tanzania. So I mentioned you about biobanking parasites. So first of all, I want to mention about why do we need to biobank parasites. So what we are focusing on is that parasites can be used as a novel materials for future biological, biomedical industries and researchers itself. For example, Trichilis suicis, which is 돼지편충. If you eat the 돼지편충, it attracts the immune system on your body, that in the process you can be cured about your autoimmune diseases such as Crohn's diseases, inflammatory bowel diseases, which is called IBD. So that you can see that parasites formerly considered as a infection to be cured, but nowadays it has to be more have a interest that can be used as a novel biomedical products. And why Tanzania? Why Tanzania do we proceed the biobanking? Is that Tanzania is a biodiversity is well preserved. The 33% of Tanzania land itself is assigned as a preserved area, so that many host species of the parasite is well preserved. So we expect that 1,922 species of hosts are abundant in Tanzania, so that its parasite is also abundant. And in the wildlife itself, the infection rate of the parasite is more than the 99%. So when it comes to biobanking parasites, the Tanzania is the best country to proceed the parasite resource biobanking. So we are doing that with on behalf of KOICA project. In the KOICA project, we are participating with the Sokoine University of Agriculture in Tanzania, and also with the Tanzania Wildlife Research Institute, which is called TAWIRI. Along with two institutions, we are focusing on the four activity. One is the education, and another thing is securing parasite resources, and third one is quality management on the secured parasite resources, and fourth thing is, most importantly, we are gonna distribute the secured parasite resources to USA, European country, or any other country who needs parasite resources to research on, to develop a novel biomedical products. So in the process, we are performing the educational program in Sokoine University to have them get to know how to identify the parasites, how to collect them, how to properly label them and biobanking them. So by training them, we can hire them in the Tanzanian branch to perform the parasite collections. So in the collection itself is stored in the Global Biobanking Hub, which is operated in Tanzania also, and Osong here in Korea. And during the intermediation, we will find the demands for the secured parasite resources to be used as a novel materials for any other researches and production development. And as far as we are starting this project since 2020, for now we have been trained 124 parasite resource experts, and we have set up 2 sites of parasite resource biobank in Tanzania itself. One is in Sokoine University of Agriculture, and one is inside of Seronera Wildlife Research Center. So funny thing is, in Seronera Wildlife Research Center is located inside of the Serengeti National Park itself. So we can collect parasites from wildlife very easily, like parasites of lions, giraffes, buffaloes, and hyenas. So we will have a set of the parasite of the specific host so that we can introduce to be used as a novel material. And since today we have collected 6,730 parasite resources. And you may wonder, then what parasite we should have collect first? So here I want to give you an example of it. One of the example is called Homunculus. So it is the name of genus, not the species itself. The life cycle of the Homunculus is it lives inside of the guts of the sheep, goats, and cattle, and attached to the gut, and they suck their blood. Eventually, the infected goats or sheep are losing weights whatsoever, and eventually they come to die. So this Homunculus infection is most abundant in Tanzania area. I believe 70% of the cattle is infected with this Homunculus. But the actual problem of the Homunculus is that the local farmers are not treating Homunculus very well. They get the aids from like USAID's, FAO's to provided by the cure, such as albendazole, lamivudine, triponite, which is a conventional treatment for the parasite infection, but they do not comply with the treatment itself. So local farmers inject the treatment, but 2 weeks after they does not do anything. So as a conclusion, Homunculus is basically get vaccinated for conventional treatments. So most of the Homunculus in Tanzania tends to have a drug resistance towards the conventional treatments. So we believe that any company wants to research its genetic characteristics on the drug resistance and have to develop alternative treatments like vaccinations and some kind of natural plant extractions. We believe that Homunculus is most demandable species in Tanzania. And we are collaborating with Professor Eenennaam, who is originally in here, 충북 National University of Medical School. By come to collaborating with him, we have come to know that treating parasites is very difficult. So one of the example is schistosomiasis, so 주혈흡충, which is the 4th dangerous parasite infection among the species, and every year 2 million people are dying from this schistosomiasis. So it is second best from the malaria. So malaria is the first, and this is the second responsible parasite infection that causes a lot of death. So this infection is very severe near the Lake Victoria area in Tanzania, Victoria 호수. Inside of the Victoria Lake, there is an island called Kome. So there are some elementary school in Kome Island, but serious thing is 97% of the kindergarteners and elementary school children are infected with the Schistosomia mansunai. The problem of the getting infection is the water get into the gut belly itself and it attached to the bladder and it causes the severe malfunction in the kidney. So eventually children are going to die. So why does this infection is too high? This Schistosomia mansunai is waterborne. So the adult worm itself is attached to the bladder and lives there and lays eggs. Then the eggs itself is come out of the urine and it is waterborne. So eggs is hatched in the water. The larva itself goes into the freshwater snails, and in the snails they can become the adult worm. So once they become the adult worm, they penetrate the human feet and get into the bladder and get infected again. So since it is the freshwater born, the children in Lake Victoria area have no system for water plumbing. They have to use the Lake Victoria as a toilet. They wash their clothes. They drink from the water from the Lake Victoria. So even if they are treated with the conventional treatment, they keep infected again. So as we come to know, to treat parasites, we have to do more than developing a new treatment or developing alternative treatments. We have to improve their lifestyle and the income itself so that they can have a sophisticated lifestyle like plumbing, those toilets, those we already have. So we are thinking of how to increase their income. So we are thinking of two ways. One is just obviously we have to first start with the agriculture production. Obviously it is more easy to start with. And there's a two aspect. So one, we go for the general agriculture like rice, potatoes, and wheat or something like that, or we can do with the livestock. But we are focusing on the livestock because once you have a cattle inside of your house, you can produce milk yearly and on and on. And even if you cannot produce milk anymore, you can butcher them and produce the meat itself. So that's why we are focusing on to proceed the cattle breed improvement projects in Tanzania. So before we starting with that, here are some few backgrounds towards the cattle in Tanzania. So Tanzania beef production is very insufficient, but the demand itself is very high. So like you can see that 80% of total consumption in Tanzania beef is imported from Kenya, Uganda, or any other European countries. And also for not only for the beef itself, the milk itself is shortage for the demands. So this is statistics for the Tanga area. Tanga area is one of the largest area in Tanzania for dairy production. So in Tanga area itself, the demands for the milk itself is 120k liters per day, but production is very shortage, so less than half of it. And the reason why the productivity is so low is that the breed improvement itself, the concept of breeding improvement in Tanzania is not well adopted. So as you can see that 98% of the cattle is just the indigenous ones. And only less than the 1% of total calves are born with the artificial insemination. The rest of the cows are just produced by the natural intercourse. And of course, the high fatal rates of cows is the problem. It is more than the 5 times in Korea. But you may say that since it is Tanzania, they do not need a high quality beef or high sophisticated milk production. You may say that do Tanzania eat cheese or yogurts or something like that? But the total, compared to the total demands, only by 5%. Only 5% is produced inside of Tanzania. So rest of the demands you have to importing from Southern Africa and Kenya. So as you can see that in Tanzania, the needs and demands for cattle improvement is very high, but the status itself is very not insufficient. So that's why we are trying to implement the new cattle breeds. So in this way. So our first priority is to have a quality breeding cattle. So in the process, we will import the semens from Korean side and do the artificial insemination inside of Tanzania side. So along with that, as Professor Rothschild just mentioned, the data management of the lineage itself is very important because we have to track and compare the phenotype data whether or not the breed itself is well improved. So we will apply our IT system to Tanzania to operate and manage the lineage data of the cattle itself. So how we are going to do that is by collaborating with the local collaborator. We have Borigaram Agricultural Technical College. As you may know, the name itself is from Buddhism. So Dreaminess, the NGO, 아름다운 동행, 조계종에서 설립한 대학입니다. 그래서 we have collaborating with this college, which have the courses for the animal sciences and livestock, and it has been established since 2016, and for the 3 years, it have 236 graduates. So it is well-working institution in Tanzania. So we are focusing on to have a center for artificial insemination and genetic analysis for the cattle breeds, so that we can produce semens for ourselves. Due to the time, so I will just skip our project ongoing outlines, and I will summarize my session by remind you about our finance business model. So as I said, we will gonna imply the IT systems and also the genetic technology to selectively choose the cattle that have good phenotypes. And once we get those good phenotype cows, we will produce the semens of ourselves in Borigaram College. Then we will distribute our semens to local farmers to share our result on the genetic improvements. And eventually, we will set up a brand production on behalf of Borigaram so that we can sell our production from meat and beef from Borigaram Technical College. So that is my session. So if you have any interest, please contact. If you want to work in Tanzania, 탄자니아에서 일하고 싶으신 분은 저희한테 끝나고 연락 주시면 한번 이제 같이 기여할 수 있는지 한번 알아보면 좋을 것 같습니다. 네, 시간 내주셔서 감사합니다.
Thank you very much. We'll have a short break from now, and we'll have a 10 minutes break. Thank you very much. 없으면 두 번째 세션 시작할게요. 그럼 지금으로부터 두 번째 세션 시작을 하도록 하겠습니다. 두 번째 세션의 첫 번째 연사 알레슨 반 에너남 교수님을 소개해 드리겠습니다. 에너남 박사님은 유니버시티 오브 캘리포니아 데이비스의 축산학과에서 동물유전체학 및 생명공학 분야 교수이십니다. 그녀는 호주 멜번 대학의 농과대학에서 학사 학위를 받았고 UC 데이비스에서 축산학 석사와 유전학 박사 학위를 받으셨습니다. 그녀의 연구 분야는 축산업 분야에서 생산성을 높이는 기술로 동물 게놈 및 생명공학의 사용에 중점을 두고 있습니다. 닥터 반 에너남 교수님은 현재 수행하고 있는 연구 프로젝트에는 소에 대한 게놈 편집 접근 방법이 포함되어 있는데 그동안 전 세계에 약 750분이 넘는 초청 연사로 초대되어서 그녀의 연구를 발표하고 있습니다. 또한 다양한 미디어를 사용해서 일반 대중에게 과학과 기술에 대해서 알리고 있는데요. 특히 GMO라든지 아니면 형질전환이든지 이런 논쟁, 거리가 될 수 있는 것에 대해서 목소리를 과학적인 사실을 전하는 목소리를 내고 있으며 특히 이제 TV에도 많이 출연해서 그런 토론 활동을 하시고 계십니다. 지난 2017년에는 아주 유명한 다큐멘터리인데요. 푸드 이볼루션이라는 다큐멘터리에 출연하셔가지고 열정적으로 과학 기술의 중요성을 말씀해 주셨는데요. 오늘 Importance of Genetic Improvement to Future Sustainability of Agriculture 축산업에 있어서 유전적인 개량이 앞으로 얼마나 중요한지에 대해서 말씀해 주시겠습니다. Dr. Alison, Benena.
Thank you, Dr. Kim, and good afternoon, everyone. Thank you for the invitation to speak here. And I think my talk is a nice complement of the previous talks and the talk that comes after me, and hopefully brings together the topic of today, which is the importance of genetics to the sustainability of animal agriculture. And if we look at these two animals, they have been selected by breeders from their ancestor, the wolf. And we've been pretty good at selecting different breeds of dogs from the wolf. Maybe this isn't really an improvement on the wolf, but certainly breeders, by looking at characteristics that they like, the phenotype, can make dramatic changes to animals. And whilst this is fun when we're talking about dogs, it becomes very serious when we're talking about our food animal selection. And shown here is selection in poultry. So what is shown is the difference between 1957 genetics and 2005 genetics at the same age, on the same feed, in the same environment. So what you're seeing there is just genetics. Breeders have selected for animals that are more productive based on their appearance and have dramatically increased the yield per bird. And that's important because it reduces the amount of inputs required to produce a kilogram of chicken in this case. And dairy cattle have a similar story to tell. And what we can see here is back in the 1940s, the average milk yield per cow suddenly went through an inflection point that was enabled not by artificial intelligence, but by artificial insemination. That is the ability to use the very best genetically elite males more widely through the use of an advanced reproductive technology. And the knock-on effect of that is not well appreciated, I think, because genetics is a key contributor to sustainability. So in the United States alone, if you look in the 1940s, there was around about 26 million dairy cows in the US. As yield increased per dairy cow, the number of cows required to produce milk for a growing US population decreased. It decreased dramatically. It went from 26 million to 9 million dairy cows today, but the amount of milk doubled, even though the number of animals decreased dramatically. And all told, what that means is because there was basically a fourfold or more than fourfold increase in milk per cow, today we only have a fifth of the number of cows, 21%, eating less feed, drinking less water, on less land, producing less manure, and ultimately the environmental footprint of a glass of milk in the United States today is 1/3 of what it was back in the 1940s. That's kind of a hidden opportunity cost that's delivered by improved animal genetics. And I think it's safe to say that genetic improvement is a powerful tool to drive animal ag sustainability. And I'm actually surprised. I thought the contract of a geneticist was you had to show the breeder's equation in every single presentation. Here's the breeder's equation, which says that the rate of genetic gain towards, hopefully, your structured breeding objective is proportional to the selection intensity, that is how widely you use the very best animals, the accuracy with which you can identify those animals, the amount of variation you have to work with to select those animals, and inversely proportional to the generation length. That is the age that parents are when their offspring are born. The more you can do to reduce that, because it's in the denominator, the more you will accelerate the rate of genetic gain. And as Dr. Rothschild explained, as we have sequenced the bovine genome, which happened back in 2009 after the sequencing of the bovine genome, we were able to look at the 3 billion base pairs that make up the double helix of the bovine genome and see that we have a lot of similarities with cattle. We're about somewhere in the vicinity of 83% identical. That 17% that differs is what makes a cow, and importantly, is the genetic variation that's available for our selection programs. And once we had sequenced the bovine genome, we didn't stop there. We sequenced thousands of bovine genomes. And what we found is the genetic diversity of all of these different breeds of cattle is reflected in the fact that there are millions, greater than 86 million genetic variations or alterations between different breeds of cattle that is reflected in single nucleotide polymorphisms and indels that makes them look different and behave differently and grow differently and be suited to different environments. A lot of the traits we're interested in, growth, marbling, are controlled by many genes. There are, however, some that are single gene traits that are controlled by single genes. And I'm going to describe one that will be of relevance to some conversation later, and that is the gene known as myostatin. So myo in Latin is muscle, and statin is stop. So if you have a mutation in the myostatin gene, you get myogo. And so you get effectively this very, very big muscle growth phenotype. This picture was taken 2 weeks ago now at a cattle show in France, where a number of the breeds are dominant mutation for this recessive condition. And you can see, just by looking at it, I don't need to genotype it, that that animal is a double-muscled animal. So because we now know that there's these millions of genomic alterations between different animals, that really enabled the development of the high-density SNP chip that Dr. Rothschild alluded to earlier, which basically enabled us to genotype in each of these little squares, little rectangles, the 50,000 different genetic loci for different SNPs, single nucleotide polymorphisms. And by marrying that to phenotypes that we had on these genotyped animals, we could get an estimate of whether a SNP was associated with a positive or a negative effect on our trait of interest. And by doing that on large reference populations, we can effectively get a genomic estimate of the genetic merit of an animal based solely on its DNA. That's called genomic prediction. And the way it works is kind of summarized on this slide. So we all know how this is how genetic works. You have a superior father, a superior mother. They produce offspring that on average should be better. But the alleles segregate due to Mendelian segregation, and some animals will get a better selection of alleles from their parents than other animals. And so if we take the best cow and bull and produce a group of full siblings, some of these got lucky. One of them got really lucky and got all the good alleles from mum and dad. How can we tell? Well, we can run their DNA on a SNP chip, and whilst we know they had good mum and dad, we can estimate that pink square here, that is the Mendelian segregation, and identify who got the very best selection. And then he is going to be a bull that goes into our breeding program. And because we know this at birth, when we can sample the DNA, we can use him as soon as he's sexually mature, at 2 years of age, instead of having to wait 5 or 6 years to do progeny testing as we used to have to do in the past. And this technology has been so widely adopted in the global dairy industry, I don't think that we could have predicted in 2009 how popular this technology would be and the value it delivers not only to elite bull breeders but also to commercial producers. And this shows the adoption and the number of genotypes that have been performed in the US with regards to dairy cattle since the introduction in 2009. And you can see there's over 6.6 million dairy cattle genotypes that have been performed. And not only is it done on bulls, shown in blue, but increasingly on commercial cows, where farmers use this to estimate the genetic merit of their commercial females, and they will breed from the top end, and the bottom end, as the lesser genetics, they will put beef semen or, in fact, beef embryos into those animals to produce a beef calf because they don't want to reproduce the genetics of that dam, and they will get a more valuable animal for the beef industry and utilize this genomic information to manage their herd differently. This has been adopted globally in dairy populations, and this is just numbers of what's been genotyped in the US around the world. So about 5 million in the United States or North America itself, but you can see it's really been adopted globally, and this doesn't count genotypes done outside of the US. And so it's a success story. We always give this as our example because it is such a success story. And what we can see here on the x-axis is the average net merit, which is basically the breeding objective for dairy cattle in the United States. And what you see is before the introduction of genomic selection, the average rate of gain, which means how much better a calf that's born this year versus last year, is about $40. So you make $40 more profit from a bull born this year than last year. That rate has doubled as a result of the adoption of this technology because it increased our accuracy and decreased our generation interval, both of which accelerate that delta G in the breeder's equation. So that's a really nice story. Why is this important? I think this is important when we look at projected demand. And so this is FAO figures. And if we look at ourselves here, we're at about 1430 million metric tons of animal source proteins. Milk, number one, followed by fish, which is perhaps not as well known, but then we see the land-based animals, the chickens, the pigs, the cattle, and eggs. And basically what needs to be produced by 2050 is around about the same amount as all of the eggs, cattle, and pigs that are produced today. So where are we going to get this additional yield from? And this is a slide that was shown earlier, showing that most of this increase is going to be happening in the developing countries, and particularly it's going to be happening in the poultry and chicken industries. So are we going to address this demand with more animals, or are we going to address this demand with more productive animals? I think that the latter is better from an environmental perspective if it's something that we can achieve in the developing world. And you often hear in the developed world that we should eat less animal source foods. And I think if we look at this graph that shows consumption of animal source proteins throughout the world, we see some interesting trends, that there are some developed countries that certainly eat more animal source foods than is needed by their dietary requirements. And it doesn't escape my notice that I'm from Australia and I now live in America, so I've always been in countries that consume a lot of animal source foods. But what you'll see on the left there is that there are countries that consume very little animal source foods, not by choice necessarily, but by lack of availability or the cost of that animal source foods. And India there on the far left obviously has some religious prohibitions around consuming beef, but they actually have a very high rate of childhood stunting due to inability to provide enough animal source foods for normal cognitive brain development. And so there is a very real need in these countries to actually supplement the amount of animal source foods available. And we often hear the discussion framed in the first world context, where we have more than we need, without considering the fact that that's not true in all the world. And I think that there has to be a more nuanced discussion around this. I'm going to look at the summary of the tremendous developments that have been made since the 1960s to the current time in our different animal industries. And I'm going to do this with a little help from David Bowie. And so what is shown here is the amount of meat per animal from 1960s through to the current day, and watch what happens around the world. This is cattle meat, beef. This is pig meat, pork. Notice it's not unique. Not all countries are growing and advancing at the same rate. This is milk. Eggs per poultry. Look at Africa. And then this is a really important one. This is why that's an important one. This is number of animals slaughtered over time. That is poultry. If we look at the number of chickens killed, it dwarfs everything else. Around about 70 billion a year. And you see way down there the number of pigs and all of the other species. So how are we going to address this increased demand, particularly for poultry and pork, where we see it coming in the developing world? And I'm going to segue now to discuss this technology of genome editing, which I think will help with the next presenter as well. You've probably heard of genome editing. There are a number of different editors available: zinc finger nucleases, TALENs, CRISPR-Cas9. What they do is introduce a targeted double-strand break into the genome, which can be repaired either using the knockout or left-hand pathway there, where you're just repairing that double-strand break and it doesn't repair exactly correctly and it inactivates or turns off a gene, mutates it, if you will, so you get a gene knockout. Alternatively, you can get a gene knock-in, where you provide a donor template And that can integrate through homology-directed repair to introduce, for example, an allele from one breed into another breed. I'm going to play a little movie that helps to explain this, so you get a rest from my voice and you hear somebody else's accent for a minute.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting unnaturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. University of California, Davis, animal geneticist Dr. Alison van Eenennaam and members of her laboratory are exploring genome editing tools that could allow breeders to precisely introduce useful genetic variations into livestock breeding programs. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual, what they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has two components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
So of course, when we're editing, we want to introduce these useful genetic variations into the germline, the semen and eggs from the next generation. We want to do germline editing if we're animal breeders. Otherwise, we haven't accomplished our goal. And in a perfect world, we probably want to introduce it in a non-mosaic homozygous condition. I'm not going to go into how that's done, perhaps the next speaker will. However, that's what our aim is. So what are some of the things that we might knock out? In other words, that left-hand pathway inactivating a gene. We might do things that would be part of a normal breeding program. Normal breeding programs, with or without genome editing, we would like disease-resistant animals. We would like products that have reduced allergens. We want to remove unwanted development. We'd like thermo-adapted animals and animals that produce more yield. And so I'm going to go through some examples of different work that's been done by groups around the world. For example, there is a knockout that results in pigs that are resistant to porcine respiratory syndrome virus, or PRRS. This is work that's been going on in the United States, in Scotland, and also in China. And you can see there the global distribution in orange of this very severe respiratory disease in pigs. I think we're all familiar with respiratory diseases at the moment. I guess as a geneticist, I think genetic improvement, permanent and cumulative, is a better solution to animal disease than having to treat sick animals with antibiotics and chemicals. Here's work from New Zealand to knock out or inactivate the beta-lactoglobulin protein, which results in some people being allergic to milk. So this milk potentially has lower levels of beta-lactoglobulin and could potentially be drunk by people that have that allergen. Work from Excelligen in the US is trying to knock out a gene associated with development through puberty in pigs, with the idea of keeping pigs in a prepubescent state so that they don't develop boar taint, which is the taste associated with meat from an intact male. Castrating pigs is an animal welfare concern in some countries, and this is an approach to avoid having to do that. Dr. Rothschild already touched on the SLIC mutation. It's a knockout of the prolactin receptor gene that basically keeps an animal's temperature about 1 degree lower than an animal that isn't mutated for the SLIC allele. Here's some work out of China that is looking at editing myostatin in pigs, cattle, and sheep. This is from a presentation given recently at a global conference on regulation of genome editing. I want to remind you that myostatin knockouts are part of naturally occurring breeding programs. As I mentioned, I was in France a couple of weeks ago. Here's a great big old myostatin knockout cow that was on display as a nice example of her breed, and it's kind of interesting to me that you can see her cesarean scar down her side there from where they've had to actually deliver her calf as she's too muscular to actually give birth naturally. So these mutations already exist in our breeding programs, and some countries have taken the approach that if you do a knockout where you're not introducing novel or transgenic DNA, foreign DNA, you're not treated any differently to conventional breeding. And specifically, those countries have regulatory policy with exclusion shown in green here. And for example, knockouts in Australia, Brazil, Argentina, Canada would not trigger any quote-unquote GMO style regulations, which opens up the opportunity to utilize those technologies in those countries. You'll notice, as of yet, Korea is in blank, which means they haven't yet determined their regulatory status. And so there are now examples of myostatin tilapia that have been judged by both Brazil and Argentina to not be GMO. And recently, Japan has determined myostatin knockouts of pufferfish and red sea bream are also non-GMO, and this is currently being sold in Japan to consumers. It's basically on the market. Similarly, cattle with simple modifications like SLIC, and in fact a double-edited SLIC/hornless Holstein has been judged by Argentina to be non-GMO, and so those products won't be treated differently to conventional breeding. And similarly, cattle with simple deletions for myostatin in the case of the Nelore animals at the top there, and SLIC animals were both considered non-GMO in Brazil in 2021. And interestingly, even the US FDA has determined that 2 myostatin knockout cows, or a cow and a bull, were also low risk, and they have been given enforcement discretion, which means that those animals are allowed to enter the food supply. Those are all knockouts. What might we knock in? So now we're going down the right-hand pathway, the homology-directed repair pathway. Again, our breeding objectives haven't changed. We still want disease resistance. We don't want unwanted development. We want thermotolerant animals. So here's some examples in China where they've produced cattle that are resistant to tuberculosis. So again, a disease-resistant application. And in fact, there's work in China to do a triple edited animal that's a myostatin knockout that is both PRRS resistant and also TGEV, or transmissible gastroenteritis virus resistant. All told, according to this presentation a couple of weeks ago, there's about 20 genome edited livestock applications in China that are going through the development pathway. Nothing on the market yet, but there's a number of different applications. And I think there's work there to address classical swine fever resistance, but the holy grail for pigs, I think, has been recently, since the spread of African swine fever to 45 countries, is there a genome editing approach that can be used to produce pigs that are not susceptible to African swine fever? Work in New Zealand to produce black and white dairy cattle that have brown and white to have less heat absorption. And then work that Excelogen did in the US to introduce basically a duplication at the allele on the pole gene to produce hornless dairy cattle. And these animals were produced, and this dominant trait, the dominant allele that results in not growing horns, was tested on by crossing horned Herefords with that bull, and Mendel was right. So none of those animals that received the horn the hornless allele grew horns, and so on the right here you can see one of the offspring of the genome edited bull that didn't grow horns and a control that came from a Holstein that did grow horns. And we have traced and followed these animals over the course of their entire lifespan and found that the growth and health and milk and meat of the 6 heterozygous hornless offspring of a genome edited bull were equivalent to their contemporary horned controls, as expected. So I think in summary, genetic improvement has clearly been an important driver of agricultural sustainability. Genome editing offers an approach to introduce useful genetic variation in alleles without the linkage drag that's associated with traditional crossbreeding. Regulators in many countries have made the decision that simple knockouts and moving an allele from one breed to another is non-GMO. It's not going to be treated differently to conventional breeding. And in my opinion, the fate of this technology will really depend upon developing a harmonized, risk-based regulatory framework that enables international trade of the products, milk, meat, eggs, and gametes derived from genome-edited animals. So I just acknowledge my funders and thank you for the opportunity to present here.
Thank you very much to make it on time. It was an excellent presentation as well. 여러분들 이제 마지막 한 개 톡이 남아 있어요. 이렇게 영어로만 진행을 하다 보니까 쉽게 그러니까 이제 받아들이기 어려운 부분들 그래서 지루할 수도 있었을 것 같은데 나중에 이런 순간들이 지금은 이해를 못하겠지만 오래오래 기억이 될 거라고 확신하고 있습니다. 왜냐하면 여러분들이 오늘 들었던 이야기들이 미래를 위한 거거든요. 그 미래가 여러분들이 살아갈 현재가 될 것이고 그리고 오늘 들었던 내용들이 그 미래에서는 해상될 거라고 저는 생각을 하고 그렇게 좀 주목을 하고 또 나중에 이제 또 이렇게 이 자리를 또 기억을 하면서 여러분들이 미래에는 좀 더 나은 우리 세상이 될 수 있기를 바라고요. 그런 의미에서 제가 이제 그 서울대학교 수의대 장구 교수님을 소개해드리겠습니다. 우리나라에서는 유일하게 한우에서 진 에디팅 유전자 편집으로 새로운 종모우, 새로운 형질을 가지고 있는 한우를 생산하고 있습니다. 그것뿐만 아니라 또 그 정액 그러니까 이제 지금 우리나라에서 정액을 농협중앙회에서만 지금 공급을 하고 있거든요. 그래서 이제 우리가 좀 더 발전할 수 있는 그런 기회를 제한적으로밖에 못 가지고 있는데 새로운 형질을 도입한 유전자 편집 기술을 사용하지 않았더라도 종모를 또 생산하고 있는 일들이 있고요. 그래서 아주 촉망받는 미래가 이렇게 장구 교수님의 연구를 통해서 만들어지기를 바라면서 장구 교수님의 발표를 들어보도록 하겠습니다. 감사합니다.
고맙습니다. 네. 네, 고맙습니다. Thank you. Thank you for invitation. I'm very happy to see Dr. Alison and everybody here. I was the bovine reproduction project was starting almost 20 years ago. When I was firstly entering this field, I was very shocked in the television because when I was in the military service, I got the television about the successful story about the cloned dollies. When I was in military service, I got in television in the future, maybe in the cloned technology will be powerful change in the reproductive including some livestock area. And then after military service, I will be entering the animal, the reproductive physiology, the clinical area, particularly embryo transfer. So basically, I'm the veterinarian. So I was some of my job was to contribute to the animal treating by clinical. So in the morning, before coming here, I was treating the two patients in the dog. So one is some estrus problem. The other one is some kind of some genetic defect in the reproductive area. So very recently, and then after getting the cloned dolly, another some big technological jump is the kind of the genetic engineering tool, particularly in the CRISPR-Cas9. I think there will be, Dr. Alison already mentioned about the CRISPR-Cas9, the properly changing the whole of the world. So in many applications will be already proved in many the scientific field. I believe so. So this one, this is the Alberta in Canada. Lastly, the previous semester I was the sabbatical year. I was visiting again my postdoc laboratory. The postdoc laboratory boss, he was the very big guy in the mouse mutagenesis consortium in the North America. Dr. Andras Nagy, he was the main leader in the mouse mutagenesis. And then after visiting I'm bringing my family to visit the Alberta to look around some beautiful the kind of the meadow like that. So, she already mentioned about the big application in the CRISPR-Cas9 studies. I think this is the big study about in dehoning the cow and then PRRS resistant. This will be future plan in the livestock application. When I starting some genome editing, I also starting some around 10 years ago. Firstly, I was exposed to the zinc finger nuclease. That will be still available in the many scientific field. But in my experience, the zinc finger nuclease is technically it's very hard, and then efficiency is very, very low. So initially, I was starting 2010 with the trial to zinc finger nuclease in the bovine and the porcine somatic cell, and then I gave up. Oh, it will be possible, but practically it is very hard to make some design the animal by editing. And then the two papers came out is the CRISPR-Cas9, and then study it. Oh, it will be possible to make some genome edited area. So this is the big impact to me, and then mostly the research is focusing the cattle. Everybody well known in the life science area, more in the cattle will be some domesticated around 10,000 years. I don't know exactly that. That will be is the changing the human breeding system. Firstly, in initial stage. Here is the artificial selection and crossbreeding, and then genetic selection starting with the human society like that. And then everybody alone the assisted reproductive technology. It was developing firstly the semen freezing. It was the powerful impact in the livestock improvement in all of the species. And then artificial insemination, in vitro fertilization, multiple ovulation, embryo transfer, and sexing, embryo sexing, sperm sexing, blah blah. Some kind of the artificial assisted reproductive technology still it has been developing in many scientists. Another little bit same one, but I was separately divided this technology, nuclear transfer. It has some advantage or even disadvantage. So right now, yesterday I was in the car, Dr. Alison, we was talking about the nuclear transfer, the disadvantage. It's powerful tool, but they have some congenital defect, sudden death. It needed high skill. It's many kind of the funding support. It was very difficult to me. And then this technology, nuclear transfer, was made another drive to scientific area induced the pluripotent stem cell. In this one, it will be supporting some human medicine in the IPS study. Everybody well known, the Shinya Yamanaka. He was developing some human-induced pluripotent stem cells for the transcription regulation factors. This technology is keep going on, and then genome editing technology is combined in the line now. And then the other some scientific field is a kind of the marker selection and some total omics, kind of the omics data will be support some more designing or editing the breeding program. Alison already mentioned about the already big change in the livestock area. Every time I was talking about some assisted reproductive technologies will be changing the milk production. This is the capturing the some USDA data. Take a look at this right now around the number of the cows is decreased. The milk yield is increased because with the semen selection and the artificial insemination and superovulation, embryo transfer, it will be changed the productivity in the milk area. So I briefly mentioned some reproductive technologies about some basically the still a very limited the farm range. They still consist about the natural breeding. They don't think about just the selection system, just the breeding. So, but initially in the industrial level of the ranch, they will be focusing the only selecting the female and male is the apparently the looks good the meal, but such as some high meat and high milk yield area. This is the still classical this breeding program is the disadvantage is the very low impactive breeding. Another one, this AI-based, the artificial insemination-based breeding program. This is the currently, and all of the culture, many country we are in Korea even we are doing the artificial insemination. The semen was collecting the higher superior the bulls, and then freezing and then inseminating the selected cow or just the cow, normal cow, and then after that F1 offspring, and then we going again selecting the some good level of the some cow, and then again another semen will be breeding. Another some technologically in the artificial insemination area, recently the time fixed artificial insemination was performed in some North America and South America country. In Korea, still it was the adjustment right now in big farm, because in detecting the estrus like that, detecting the estrus like in the mounting area and kind of the some discharging about it, it will be such a pain in the farm. So based on the some hormone control, they inducing the time fixed the estrus. So they didn't detecting the estrus. Just go to the veterinarian or technician. They visited the farm, only the insemination without any the estrus detection. Look at this. The graph here is the time fixed artificial insemination. Time's going on, and then practical level is going on. So nowadays, around the 70 or 80% in AI farms, they'll be choose some time fixed artificial insemination, and then another challenging to make some sire and them to based on some multiple ovulation embryo transfer breeding program. There will be some technically many challenges that we have to make some super ovulation hormone program. In another one, we have to selecting some male ones and then inject and collecting some high viable embryo and transfer the recipient. This the multiple ovulation and embryo transfer is still many countries. This is the best choice for high pregnancy and then make some sire and dams. This technology have the pros and cons. Pros is the high pregnancy. Cons is the improving the phenotype, and the cons is the higher skilled and then cost. And skilled person in need the 2 or 3 years of training to manipulating the uterus, and then insemination, collecting, handling, and transfer. And in another challenging one is about the OPU-based embryo transfer nowadays. Previously, hormone-dependent embryo transfer, it means it needed some technical issue about that. Even this the OPU-based one is the same. It's the same. We have to based on the time to learn the manipulating the OPU, ovum pickup technology. We need the big money to buy the ultrasonography. High resolution ultrasonography is the price about around the 5천만 원. $50,000, over than $50,000. We needed some more extra device to handling the oocyte collection. Anyway, after establishing the ovum pickup system, and then the other one, in vitro fertilizers, it was very well developing all over the world, and we can easily get the in vitro fertilized bovine embryo and transfer the recipient. This is the pros and cons even. Pros is the high pregnancy. After 2010 years later, is a high pregnancy. The low pregnancy is before the 2010 is a low pregnancy. It depends on the embryo culture media and the embryo transfer technology one. This is some kind of the comparison of the in vivo, in vitro embryo. In vivo means the ovulation. In vitro is OPU based. This picture is about the data collecting from International Embryo Transfer Society, and then from the from the 2013. This the number of the in vivo embryo was decreased like that, and then in OPU based or slaughterhouse in vitro fertilized embryo was production is high increased then. 2021 it was the 3.4 times more productive based on the OPU based one. Mostly in the North America and South America, there will be leading country to make the OPU based one. This one will be more powerful in practical one. In Korea, even some of the company, also even my laboratory, we are starting the already OPU based program. So compared to other species, the cattle is the long generation interval, low fecundity, and slow genetic progress. So still, many scientists and farmers, they'll be one of the interesting areas is to how to make the sire and dam level. This is I'm thinking about the breeding program with the genetically engineered tools. So firstly, we have to selecting the donor selection, either female or male, and then one of the approaches the in vivo embryo, and the other one OPU based one. In OPU based one, it should be involve the in vitro production embryo. We have to collecting some high transferable viable embryo, selecting the viable embryo, transfer the recipient, and then this recipient is delivered some F1 the delivered elite cows. This is gross, and then again selection is based on the genome selection. And then again, the female and male will be donating the male and female. This circle will be circulating. It's much more accelerating some future breeding program to make the sire and dams. I was introduced the previous work just based on this program. I'm starting making this strategy. So initially, I was my colleague, we asked together initially focusing to establish the MOET, is the multiple ovulation embryo transfer in Korea. Our group, it was starting around 20 years ago. Firstly, after artificial insemination, our group, mostly many scientists, our group is focusing only embryo transfer, multiple ovulation based one. Artificial insemination, nowadays the general person, general technician, general veterinarian, they can do everything. But in the multiple ovulation embryo transfer, it needed a high skill, and then we needed some special scientific advance then. At the time, we was around 20 years ago, we was only focusing the multiple ovulation embryo transfer area. And then after successfully make the multiple embryo transfer, our group is to move to the in vitro fertilized one. Because the multiple ovulation transfer, it needs the high cost, time waste, high money. So there is some disadvantage. So we was firstly, the scientific area reduced the price of some cultured media. Now there's one cultured media, one buyer in commercially available around the $300. But in my laboratory, we was developing some chemically defined media. One is the $1,000. So we was previously 2007 we was published about the chemically defined media. This is the time-lapse the recording for embryo development based on the our culture chemical defined media. Take a look at the beautiful the embryo development. We transfer the only selecting the some viable embryo a bit around, over than 16 stage cell. Around the 80% there will be grown up the blastocyst level, and high quality blastocyst cell level was frozen and sowing. Look at this, the time lapse recording after sowing and survival in frozen the in vitro fertilized embryo. Over than 90%, most of the embryo was survived in vitro. So, and then I was established this area, and then I moved to the genetically engineered the area tools. When I was postdoc, I was mentioned about my boss was about the reading scientist in the genetically engineered mouse. So I was at that time I was exposed to many kind of the genetically engineered tool. I was very interested about using the several kind of the genetic tools to apply to the livestock. So when I came back, something overlapping. Firstly, I was this one. Sorry, this is some representative picture about the first generation GFP whole body expression transgenic cattle. It was published 2016. It was projected starting the 2013 project about that. He is now 9 years old. He was still survive on the still GFP expression is keep going without any silence. And then I was reporting the second paper about the transgenic cattle, the long-term health and germline transmission in transgenic cattle following the transformer-mediated gene transfer. Based on the previous report, we was the 2 transgenic cattle, female and male, was natural breeding. We generating the F1 generation. And based on the whole genome sequencing, we analyzed the genome's instability, copy number variation, and structure variation. Some my colleague, they will be analyzing the whole genome, analyze the genome stability level. They talk to me, there will be everything, the genome stability is everything fine. So this F1 is still survive. His aging is now is 5 years old. And then we collecting the semen from the F0 foundation, found animal, and look at this, the beautiful the GFP expression in blastocyst level. So without any somatic cell the technology, we can make the transgenic, means the GFP long-term stable expression blastocyst was produced in vitro. And then finally, recently I was sacrificed the incinerated one. Before the incineration, I was taking the whole body, the heart, kidney, ring, lung, and then the testis. The testis, look at this epididymis area. So GFP expression well observed that. So I'm close up. This is the epididymis area. So epididymis is very important. Reproductive organ to storage and matured in the semen. That's why the GFP expression very well. And then another paper, 2 years ago, I was published about the biology of reproduction, about the F2 generation area. So keep going to monitoring the previous found animal, or it was the female and male, and then published the first F1, and then another F1 we was generating by embryo transfer, and then isolating the somatic cell and sperm. And then here, we was show some another scientific application for the CRISPR-Cas9. In based on the CRISPR-Cas9, one of the hurdle is to make the efficient guide RNA and some transfection systems. So here we was the 2 guide RNA. One is the GFP, and the other one is the target locus. So simultaneously, we can transfect together the GFP guide RNA, one the other guide RNA, and then we only sorting about the only GFP negative one. It means the GFP guide RNA working well, so that's why the GFP signals disappeared. And then we analyzing the GFP negative the cell, another target locus. It was the here is the improved around 2 times was increased. So based on the nowadays we are screening the whole guide RNA, this GFP expression cell line. The firstly guide RNA for GFP, one target locus. So it will be we called the enrichment system, means the enrichment is the increase the number of the population in the target locus. And then finally, the recent upgrade about the genome engineering one is the genome editing one. Kwan-Suk already, Professor Kim Kwan-Suk already mentioned about the successful story about the myostatin knockout HANU. Previously, by starting the genome editing in Thailand, we was applied to the porcine cell, and then another story was applied to the bovine, the prion gene locus to efficiently deleting some in somatic cell level. Based on some many skills and improving some laboratory one, we was finally made some guide RNA for MSTN in HANU, and we was targeted the exon 2 area. So we screened several guide RNA. Every guide RNA showed some high efficiency. It was very confused which one will be effectively or some efficiently produced some knockout, MSTN knockout HANU. So we was starting in already some China group based on the Thailand. There will be and zinc finger. They focusing the exon 1. The other group is in UK group. They strategizing the similar one, while the naturally occurring one exon 3. So nobody doing the exon 2 targeting. So we choosing the one of the guide RNA was which one was selecting for exon 2, and then we made some CRISPR and RNA injection in zygote and made the transfer the embryo, and finally we got some biallelic 99% knockout the female HANU. They will be grown up 2 months stage. This is already 3 months ago was the take a photo, and now it's already 28 months. Is take a look at this outgrowing muscle, not double muscle, just outgrowing. It's a very interesting one. Another one we was keep going on, and we analyzing the germline transmission, MSTN HANU, and this one we have already produced the female and male. The female, in case of female, that was employed the OPU and collecting the oocyte and fertilized together, and we got the viable embryo and transfer, and then finally we got a F1 generation. We got a 2 F1 generation line now. In the paper, one is just introduced only one, and then this semen, and then applied the wild type inseminated together. At that time, it was another transfection is kind of the electroporation. Dr. Alison already mentioned, published about electroporation is much more practical and easy. So we are same result with Dr. Alison about the electroporation based knockout one, and then we keep going to in the base one. And I didn't publish about some more analyzing about myostatin knockout HANU. We collecting some biopsy in the tissue, and then HAE staining, and then we analyzing the pixel level, every muscle fiber. Is a knockout. Muscle fiber was very thin, and then we culturing the myoblast is some muscle stem cell in the knockout HANU. This MSTN knockout one without any triggering the differentiation is much easily the differentiates the muscle fiber in vitro level. And this is the wild type one. without signal, there will be very slowly differentiated the in vitro in the muscle fiber, the type. So I believe that the muscle will be donated. We already starting some collaborated with the cultivated meat company to donating the muscle myoblast to efficiently making the cultivated meat. I don't know what in the future what's happening there. This is some general data in the published about that. And then the ongoing project about still I didn't publish about the PRMP knockout one is for mad cow disease. It was starting around 8 years ago. I was granted from the National Research Foundation to generating the prion knockout cattle. So 3 years ago, we successfully generating the female and male one. And female one, we was collecting the OPU, I previously mentioned the one, and then male one, we was subjected about the semen collection. We was the in vitro fertilized based on the knockout semen. Is look at this. Each process is to show the heterozygotic knockout one. So we believe that this 24 years old the male one is the bioallelic, almost 100% knockout one. So I'm waiting the F1 generation because the November was the expected the pregnancy the delivery date. I will be happy to visiting some farm to get some F1 calf. Another one double knockout strategy. So we was also CRISPR-Cas9 applied to the myostatin and beta-lactoferrin. Both guidelines together simultaneously the electroporation. In embryo level, we got over than 50% efficiency. We selecting the embryo was transferred recipient. Finally, we got around 10 knockout double knockout cattle. We are on the still the deep sequencing the efficiency, how much was the indel mutation find the double knockout one. And then another one is it was projected almost 10 years ago starting the supportive of the Seoul Milk Corporation. So we was make some Cas9 overexpression cattle. You know, every time technologically we have to knock out the gene, we need the two components. One is the Cas9 protein, the other one is the guide RNA. So Cas9 protein is not changed. So I'm thinking that our laboratory, we have to make some Cas9 overexpression whole body one, and then if we can successfully make some oocyte in the semen, after that we get to only guide RNA, another one template DNA, much more easily generating another one our future design in the cattle. So it was starting 10 years ago. 2014 we was made some F0 foundation male and female, and then we made the F1 generation, and then again we just collecting the semen. Now we starting analyzing the F2 embryo, the second editing and knock-in one. We have the some F1 generation Cas9 overexpression cell. Two is the dairy cow, one is the beef Hanu. So after 2 or 3 years later, I believe that the Cas9 overexpression germline cell will be powerful change in the livestock area. Another one is this is the rat project. In my laboratory, mostly research is focusing the bovine, but some of the energy was relating some basic science. One is the rat, because the rat is a little bit bigger than mouse, so we can manipulating and then much more easily translating the disease. So my collaborator, they got some project about some asclerosis. This is the coronary disease in a big issue in the human. So they needed the triple knockout rat. So we already initially made some ApoE knockout, LDL knockout. Another one, the cholesterol knockout one. So we already generating the single, and then we was breeding in the triple knockout one. One interesting finding in the CYP cholesterol knockout one, there will be some there will be sex reverse was found there. So because the other some ApoE and LDLR, I'm not interested in one because I am not the scientist about some lipid metabolism. I'm the reproductive one. Here is the we see in the gonad was the rebirth. Apparently it's the female, but inside there will be gonad testis was growing. So we are still investigate what that kind of the change was in rat. And we also to sharing the rat resource. In Korea, nobody interested about the freezing the system in the rat. So the bovine we have some many experience the bovine embryology. It means the freezing technology was applied to the rat. We can successfully generating the rat, the embryo, frozen and sowing system. All of the species, the knockout breed was frozen in the liquid nitrogen. So maybe next year I will be that kind of the rat embryo will be donated the Korea FDA to more application the other scientists. And then nowadays another big dream in my some collaboration in the Gyeongsangbuk-do. We have already made the special breeding farm here. So here is the OPU center, some space facility and microscope. Here is we already starting some genome sequencing, the superior one, inferior one, and then based on that, two comparison of the whole genome sequencing about that, which one will be like it already mentioned about some genotyping about that. Based on we maybe you got us to sharing the significant meaning of the target locus. I'm gonna do changing some gene editing our system. Maybe hopefully this breeding center contributing in the world the livestock efficiency. This is summarized one. So artificial reproduction technology. So basically still we are keep going the elite cow and bulls and introduction new breed, and previously not much contribute right now cloned cattle, and mostly ART technology was combined the genome editing one, then product disease, prion disease, product protein MSTN, and the allergy free GGTA1, MPLG1. GGTA1 is one of the glycoprotein. There will be recently in FDA, United States FDA is to approve the GGTA knockout the pig for food chains. So I hope we already also generating the GGTA knockout the cattle, and then another we in the laboratory level already proved the sleek, and then we keep more focusing some biomedical recombinant protein in the milk. So nowadays our partner we focusing some extracting the exosome from the milk. So we're gonna changing some milk exosome by genome editing one. It technically already proved that. I hope so. Some of the milk recombinant protein combined the genome editing one will be one of the application is the biomedical one. Thank you for time is over. Sorry about that. Thank you attention. My laboratory, I am founding the company together, and then the founder company the collaboration. And then I'm very special thanks to Seoul Milk Corporation and Gyeongsangbuk-do and many some companies supporting my research. Thank you for your attention.
Thank you very much for the nice presentation. 제가 잠깐 소개하기면은 우리나라의 이제 육종하고 번식이 이렇게 협력이 잘 안 되고 있어요. 그런 부분을 이제 앞으로 좀 극복하는 게 우리나라 축산 발전에 도움이 좀 될 거라고 생각이 들고요. 유튜브 채널로 그 화면 바꿔주시겠어요? 안 돼요? 여기서? 인터넷 연결이 안 돼 있죠. 저기에. 네, 알겠습니다. 그 유튜브 채널이 있거든요. 지금 라이브로 나가고 있고, 그 채널 그 여러분들이 접속을 해서 제가 여기에 그 링크를 잠깐 올리고 잠깐만요. Q&A 세션을 하려고 하는데 유튜브 채널에 거기에 이제 그 질문을 올려주기를 바랬어요. 제가 그 네 분의 오늘 연사분을 모시고 그 심포지엄을 진행을 했고, 그리고 이제 여러분들한테서 궁금한 점이 있으면은 그 궁금한 점에 대해서 답변을 할 수 있도록 하겠습니다. 네, 여기에 지금 유튜브 채널이 나왔는데요. 여기 링크 보여주실 수 있나요? 링크는 안 돼요? 여기다가요? 네. 네, 네, 링크를 보여주시면 네, 감사합니다. So we're gonna have a Q&A session from now. I'll have like 20 minutes for discussion. 제가 이제 여러분들 여기 학부생으로 계시니까 오늘 먼저 두 분이 발표한 대학을 소개를 해드릴게요. 아이오와 주립대학교에는 축산학과에 학부 학생이 몇 명 정도 될까요? 자, 1000명이 넘는다. 학부 학생 1학년부터 4학년까지 1000명이 넘는다. 500명이 넘는다. 200명이 넘는다. 자, 1000명 1번, 2번 500명, 3번 200명. 자, 1번 한번 손 들어 보세요. 아무도 없나요? 네. 자, 500명이 넘을 거다. 학부 학생이 500명이 넘을 거다. 200명이 넘을 거다. 손 들어 보세요. 네. 답은 1번입니다. 학부생만 1100명입니다. 아이오와 주립대학교 축산학과의 학부생만 100명입니다. 1100명입니다. 그러니까 한 학년에 250명에서 300명 정도 돼요. 한 학년이 250명에서 300명 정도 돼요. 우리나라 전체 축산학과에 졸업하는 졸업생 수, 전체 축산학과 졸업생 수가 한 400명 정도 되거든요. 자, 그러면 이제 제가 두 번째 대학, 캘리포니아 대학 데이비스 캠퍼스의 축산학과 학생이 1000명이 넘는다. 1번. 500명이 넘는다. 2번. 200명이 넘는다. 3번. 자, 1번 손 들어 보세요. 2번. 3번. 네. 한 학년이 800명 정도 돼요. 한 학년이 800명이 아니라 전체가 한 800명 정도 돼요. 그러니까 이제 두 대학을 합치면은 매년 배출되는 학생 수가 2,000명, 그러니까 이제 총 2,000명이니까 우리나라 두 대학을 합쳐도 우리나라 전체 배출되는 학생 수만큼 축산학과 학생들이 졸업을 하고 일자리를 구하고 있어요. 그만큼 산업의 규모가 차이도 있지만 규모 면에서 굉장히 차이가 난다라는 거. 그리고 여러분들이 그런 쪽에 좀 더 이제 어떻게 공부하고 어떻게 변화하고 있는지에 대해서 조금 더 관심을 가지면 좋겠고, 그런 의미에서 제가 이제 한 가지 질문을 드릴 건데, 아이오와 대학하고 캘리포니아 대학에 각각 다른 질문을 드릴 건데, 첫 번째, 아이오와 대학에는 어떻게 그렇게 ODA 사업을 아이오와 대학에서 관심을 가지게 되었는지를 먼저 질문을 드리겠습니다. I have one question, Max. How you become and the university were interested in the project for developing countries? I know Iowa State University has a center and a program helping livestock industry for developing country, and where the money coming from.
So it's the interest in international work is that we have a new major called called GLOBE, and this major is to introduce students to be more interested in activities around the world. We had a major donor push that, and our College of Agriculture is determined that every student will have or 3/4 of their students will have an international experience in the next 5 years. So the college made an effort to teach students to be more than just from Iowa and more interested than just the U.S. and have a global perspective. So private money and state money together help fund this effort.
Thank you very much. Thank you very much. 제가 간단하게 이제 이 부분을 소개를 하면은 미국 축산학과, 특별히 이제 아이오와 주립대학교 축산학과 학부생들은 한 학기 동안 해외에 가서 인턴십을 하는 것을 장려하고 있고 지원을 하고 있습니다. 그래서 학부 다니는 2학년부터 보통 한 2학년이나 3학년 자기가 이제 해외에 가서 공부할 기회를 가지게 돼요. 그런데 그런 공부할 기회를 회사에서 그 지원을 하거든요. 그런 것들이 이제 좀 부럽기도 하고 잘 되어 있고 그러다 보니까 자연스럽게 이제 그 해외 농업, 축산업에 대해서 관심을 가지고 또 이제 개발도상국에 대한 기술 지원도 하고 있는 걸로 알고 있고요. I have another question. I like to ask Alison. So we talk about the importance of biotechnology for developing world, and I really think Korea can be part of it. So this is your first time being in Korea, okay? And you heard about Janggu's talk, and you seen the Seoul different place. So my question will be, what is your impression being Korea, and what role Korea can play for the developing country? So you understand my question?
Sure. So this is my first time in Korea, and I had no preconceived notions what the agriculture would be like here. So I think what was interesting to me coming from Australia, which is a very pastoral system of cattle rearing, I was noticing that a lot of the animals here are inside in sheds, which was I wasn't expecting to see that. So that was interesting, and I think the level of the vegetable cultivation all being indoors in the glass houses, for one, kind of plastic houses, that's very sophisticated. It reminded me a little bit of the Netherlands in how they do a lot of their production that way. So obviously Korea is able to feed itself and has a lot of agricultural production. And also the technical expertise that as was just discussed, is doing a lot of the work. I think one of the biggest problems, as was alluded to by Dr. Rothschild, is the misinformation around biotechnology is really problematic in terms of utilizing it in agricultural production systems. And I think we've seen during COVID how misinformation can get the incorrect ideas out into the public. And I think that's happened with biotechnology, which has a really safe track record of 25 years. And I think that countries that have used these technologies and people that use these technologies have to speak out around the importance of not only biotechnology, but genetics in general, in terms of the sustainability of ag production systems. And if we want to be able to use these technologies, we have to get over this misinformation around the technology. And I think we have a duty to help do that. And I know we were speaking yesterday about efforts to make this technology understandable for the general public with videos like what was in my presentation, and connecting with where this technology can help address things like micronutrient deficiencies and produce disease-resistant crops that can help with food security. And if we need to get that message out there, because I do think it's a useful tool. It's not going to solve world hunger, but it should be able to help with things like disease resistance, and that's, I think, our global challenge.
Okay, thank you very much. 제가 간단하게 요약을 해서 말씀드리면은 우리가 이제 우리 환경을 보다 보니까 우리가 가지고 있는 장점과 단점에 대해서 잘못 이해하고 있는 경우가 많고, 그리고 이제 그런 여론이 잘못 형성되는 것도 있어요. 알레슨 교수가 언급했는 게 뭐냐 하면은 우리는 이제 가축을 키우면은 밖에 방목을 해야 되고 또 이제 뭐 자유롭게 이렇게 해야 되는 거 아닌가. 그리고 이제 그런 생각을 가지게 되는데, 그런데 실제로 이제 우리 한우 사육 농가의 그런 환경들은 굉장히 이제 동물 복지 측면에서 괜찮은 편이고요. 그리고 사용 관리나 이런 것들이 아주 집약적으로 기술적으로 많이 발전된 형태라는 것을 말씀드리고 싶어요. 그래서 축산에 대해서 부정적인 이미지, 공장식 축산이라는 것이 보기에 따라서는 이제 안 좋게 보일 수도 있는데, 이 서구인의 시각으로 봤을 때는 저렇게 하기가 얼마나 어려운데 이런 기술적인 발전을 이루어냈는지에 대한 경이로움을 또 얘기를 했었었고요. 그리고 생명공학이라는 것들도 문제를 해결하는 도구가 되는데, 생명공학을 어떻게 활용하느냐, 생명공학이 줄 수 있는 여러 가지 혜택들을 우리가 좀 더 문제를 해결하는 쪽으로 활용되면 좋겠다. 그런 부분은 이제 우리가 이렇게 많은 피해를 받고 있는 것이 이제 질병 문제, 전염병 문제 이런 것들인데, 이런 거에 대해서 조금 더 관심을 가지고 활용되면 좋겠다라고 얘기를 했었고, 그런 부분에도 저는 공감을 하고 있거든요. 우리가 가지고 있는 좋은 기술을 헛되이 낭비하고 있는 게 아닌가라는 생각을 좀 하게 됩니다. 장국 교수님 이제 서울대학교에서 그 제가 한 가지 하면은 우리 축산 분야에 이렇게 노력하고 계신 교수님 중에 한 분이세요. 많지 않잖아요. 그래서 수의대에서 바라보는 그러니까 축산에 대해서 이렇게 바라보는 연구를 하면서 느낀 점, 그 부분 우리 축산학과 학생들한테 말씀해 주시면 좋을 것 같습니다. 네, 부탁드립니다.
네. 뭐 저는 뭐 수의학과에 있다 보니까 제가 축산학과 강의를 조금씩 합니다. 베이직한 부분들을 조금 강의를 하는데, 이제 축산학과는 목적이 약간 생산성과 복지 쪽에 가깝고, 저희 수의학과는 질병 예방과 치료 쪽에 가깝기 때문에 약간은 좀 거리가 있는 편이기도 합니다. 저희 서울대에서도 썩 사이가 좋을 때도 있고 나쁠 때도 있는데, 같이 좀 생산적인 일을 하려면 도전을 해야 되는데, 이 저희들 쪽에서는 질병을 좀 다루는 입장이다 보니까 목장 입장이나 생산하는 입장에서는 좀 거리감이 있을 수 있거든요. 근데 조금만 서로 이해를 하면 결국은 질병의 방어 기전이나 질병의 발생들이 결국 T셀과 관련된 모든 면역 세포들도 관련 보면 결국은 대부분이 다 이 생산성과 다 관련이 깊습니다. 그래서 저도 이런 연구를 하는 데 있어서 서울대 목장에서 연구를 못했던 이유가, 일부 서울대 목장에 계시는 교수님들이 제가 매드카우 디지즈 없는 소를 만들겠다 이랬는데, 이거를 거기서 마치 이게 여기서 광우병을 생산해 가지고 뭐 연구하려고 한다라는 오해를 대면서 제가 목장 아예 자체의 연구를 콜라보레이션이 안 되더라고요. 근데 그거를 그 교수님은 전 이해가 됩니다. 왜냐면 질병에 대해서 잘 모르시니까. 근데 조금만 제 얘기를 들어보시면 저희가 하는 이 지노믹스 베이스드로 하는 디지즈 리지스턴트라든지 이런 것들이 지금 이해하셔가지고 저한테는 좀 미안하다고 하셨는데, 근데 한 3년 전에 제가 이 프로젝트를 시작할 때만 해도 굉장히 힘들었었던 부분이 많습니다. 그래서 이 질병과 이 발생과 환경 이런 부분도 굉장히 중요하고요. 그리고 저는 지금 현재 평창 그린바이오 캠퍼스에 원장을 하고 있습니다. 저희 산업동물연수원 원장을 하고 있는데, 저희들이 지금 수의학과 학생들을 매년 한 500명을 기본 라이브스탁에 대한 베이직을 가르치고 있습니다. 근데 좀 다른 축산학과 사람들도 좀 했으면 좋겠다라는 요청이 올해부터 들어오기 시작했습니다. 그래서 나중에 혹시 기회가 되면 뭐 충북대학부터라도 그쪽에 오시면 숙소도 다 있고요. 그래서 기본적인 삶이 되니까 오셔가지고 1박 2일로 저희가 운영하는 게 이제 1박 2일 1일 그 축산 체험이 있어요. 저희 수의학과 학생들한테도. 그래서 하는 일이 목장 가서 똥 치우고 밥 주고, 그리고 질병 공부도 가르쳐 주고, 또 이게 그 옆에 있는 동물들 보기도 하고 이런 것들 하고 있거든요. 근데 최근에 이제 축산학과도 질병 때문에 현장에 나가는 게 굉장히 제한이 있다고 들었습니다. 그래서 그러다 보니까 이 축산학과에 계시는 분들이 좀 라이프스탁이 좀 많이 안 있고 약간은 좀 다른 쪽으로 많이 가시는데, 와서 이렇게 한번 체험해 보고 계시고, 아까 우리 아이오나 데이비스나 이렇게 사람이 많았던 이유는 이 확장성이 높기 때문입니다. 그래서 한번 이렇게 잘 콜라보레이션이 되면 저희 평창에 있는 산업동물 임상교육연수원에 대해서는 뭐 저희들이 수정사도 강의도 하고요. 일반인 특강도 가끔 합니다. 관심 있는 사람들 오시면. 근데 이제 학생들도 오셔가지고 기본적인 뭐 소나 동물의 해부학도 배우시지만 다시 한번 저희과 교수님들한테 한번 이렇게 들어보시는 것도 좋고 오셔가지고 소의 어떤 기본적인 질병의 발생이라든지 그리고 저희 서울대 목장도 이렇게 한번 체험해 보시고 그러면은 앞으로 좀 더 이 지노믹스 어떤 관련된 거와 그 다음에 프로덕티버티 그리고 디지즈 이게 잘 결합이 돼가지고 굉장히 앞으로 갈 길이 많은 일들이 여러분들한테는 더 미래가 있을 것으로 생각을 합니다. 이렇게 딱 눈앞에 보이는 목장 뭐 이런 것만 생각하지 마시고요. 이걸 통해서 갈 수 있는 길이 되게 많이 있다는 것을 이렇게 아까 현장 체험을 통해서 이루어지는 것처럼 이런 기회가 좀 앞으로 있었으면 좋겠습니다. 네, 감사합니다.
네, 감사합니다. 특히 또 충청북도에서는 한우 산업 발전을 위해서 많은 관심을 가지고 투자를 하고 있거든요.
충청도요?
그리고 충청북도에서
북도에서, 네.
네, 그래서 한우 개량센터 같은 것들을 지금 설립하고 그리고 거기에 또 이제 저기 업체 이티바이오 정영길 대표님 그동안 많은 일들을 그쪽에 관련해서 해오셨는데 관여하고 있고 또 같이 협력할 수 있는 기회가 또 앞으로 마련되기를 또 바라고 있고요. 마지막으로 이제 한 분 더 이제 제가 질문을 하고 연사분들한테 여러분들 여기 이제 그 링크가 있는데 링크에 접속을 아직 못 하셨거나 하지 않으실 것 같기도 한데 꼭 접속을 해서 여러분들 소감을 오늘 있었던 행사에 대한 소감을 남겨주기를 바라요. 제가 그거를 이게 수업의 연장으로 제 수업을 듣는 학생들은 수업의 연장으로 참고를 해서 보너스를 이렇게 학점에 혜택을 주려고 하고 있습니다. 그래서 여기 링크에 꼭 참여해서 댓글 남겨주시면은 고맙겠고요. 또 이게 이제 있으니까요. Last question I like to ask Hee-Jae Yang. You don't have a background in animal science or this biology, and while you are working in this area, what would you give some comment, your impression to the student? 제 질문 이해하시죠?
네,
네, 얼마나 쉽지 않았을까라는 것을 저는
네.
느끼고 있거든요.
네, 네. 그래서 저는 지금 주식회사 코쿤이라는 회사에 지금 5년째 같이 함께 하고 있고요. 저는 교수님께서 말씀해 주시는데 교수님하고의 인연은 이제 4년 넘게 돼가고 있는 것 같습니다. 그래서 교수님께서 이제 한우 유전자, 육량 증대나 육질 증대 관련해서 유전자 분석 연구를 하신다고 하면 저희는 이제 그 종축계량협회나 이제 축협에 이제 데이터가 워낙 잘 되어 있으니까 그 혈통 정보 가지고 우량 암소를 찾아내는 이력제 정보 가지고 우량 암소를 찾아내는 데이터 분석을 좀 병행하고 있고요. 그래서 저희가 데이터로 찾아내면 이제 교수님께서 유전자 분석을 같이 검증하는 그래서 이런 사업들을 함께 하고 있습니다. 교수님께서 질문 주신 거에 대해서는 이제 축산학과에 계시지만 이제 장구 교수님께서 말씀 주신 것처럼 이제 뭐 농장이나 생산성 이런 것만 집중하실 게 아니라 이제 다른 분야와 계속 접목하고 하다 보면 저는 통계학이지만 이제 이런 분야에 지금 사업을 하고 있는 것처럼 하시는 분야 이제 공부하시는 분야뿐만 아니라 다른 분야하고 접목을 하면 이제 그 파급 효과를 더 크게 키우실 수 있지 않나라는 생각이 있고 저도 그래 왔으니까 그래서 뭐 힘든 점은 없고 이제 나아갈 목표가 있다면 그거에 맞춰서 이제 학생분들이시니까 열심히 노력하시면 좋을 것 같습니다. 감사합니다.
고민하는 것들이 무엇을 할 것인가라는 거거든요. 무엇을 할 것인가. 저는 여러분들이 앞으로의 삶에 있어서 우리가 달라진 세상을 살 건데 무엇을 하고 싶은가에 대해서 좀 더 관심을 가지고 오늘 행사가 이렇게 새로운 분야도 있구나. 그리고 또 새로운 분야뿐만 아니라 새로운 곳 그러니까 아프리카 같은 곳은 아직까지 축산이 굉장히 열악한 환경이에요. 그동안 우리가 가졌던 경험과 기술들을 여러분들이 숙지하면은 풍부한 기회가 일할 수 있는 기회, 하고 싶은 의미 있는 삶을 살 수 있는 기회가 될 거라고 생각을 하고요. 아까도 말씀드렸지만 어딜 가나 한국인이다라고 하면은 전 세계에서 가장 신뢰받는 사람으로 저는 느껴지고 있어요. 그래서 우리 뭐 한국 사람으로서 뭔가를 한다는 것은 다른 나라의 어떤 분들이 이렇게 일을 시작하는 것보다 어드밴티지를 틀림없이 가지게 되니까 그런 기회도 여러분들이 지금 느끼고 잘 활용하면 좋겠어요. 질문이 혹시 있으면은 질문을 받고 없으면은 마칠 건데 질문 혹시 있나요? 혹시 있나요? 질문이 있을 것 같지 않으니까 지금 당장 오늘은 마치도록 하고 여러분들 그 유튜브에 그 댓글로 여러분들의 소감을 남겨주기를 바라겠습니다. 네, 많이 힘들었을 건데 장시간 수고했고요. 우리 또 좋은 발표해 주신 네 분의 연사분들한테 박수로서 감사의 표시를 하면서 마치도록 하겠습니다. 네, 감사합니다. 네, thank you very much for the nice presentation.